Hi everyone. So today I'm going to present to you the general overview of microfluidic technology developed at Lunaphore and the autostainer lab set. We're going to go through a specific application of LabSat, which is the automated TSA-based multiplexing, and see how to optimize an assay on tonsil tissue. And then we're going to see a method for easy transfer of those optimized protocol to cancer tissues, and in a specific case study on non-small cell lung carcinoma. So a Lunaphore core technology is based on a microfluidic chip called MTP for microfluidic tissue processor. It is used for tissue analysis assays like immunofluorescence or immunohistochemistry. The chip is equipped with a series of microfluidic channels used for reagent delivery. It is clamped against a standard glass slide that makes a closed chamber over the tissue sample. This technology is called fast fluidic exchange for FFX. So the pressure-driven reagent flows directly, almost instantaneously, in the chamber. The liquid exchange is very rapid between the steps. A heating element placed just below the slide allows precise temperature control at any time, as well as rapid temperature changes. The perfect sealing between the chip and the slide offers a controlled environment to the tissue with multiple benefits. Staining times are much faster due to reduced diffusion times in the chamber. There is a more uniform staining over the tissue. The rapid and high temperature epitope retrieval are made possible while preventing reagent evaporation and preserving the tissue. This technology was born at the Swiss Federal Institute of Technology in Lausanne, and the efficiency of the FFX method in the IHE techniques was first shown on HER2 stainings of breast carcinoma, where it was helping reducing significantly the 2+ borderline scorings that are considered as ambiguous readouts. In immunofluorescence, the MTP-based assays were also shown to correlate with the number of HER2 gene copy numbers. The MTP chip is integrated in an automated device that is called LabSat Research. Reagents are simply loaded in 8 standard Eppendorf, and buffers are stored in 4 Falcon tubes. The chip is placed in a staining chamber maintained by a lid, ready to be clamped against a slide. Another important microfluidic element of the system is the distribution chip. It acts as a gateway between the reservoirs and the chamber and ensures the proper control of the flow towards the tissue. Reagents are pushed from the reservoir towards the distribution system, from where their dispense is controlled to the staining chamber. Once the reagent is finished, reagents are simply discarded to a liquid waste. After having tested and proven the efficiency of the MTP on single markers immunofluorescence and immunohistochemistry, we decided to move towards multiplexing assays. There is an important drive towards multiplexing techniques, as the context of personalized medicine requires even higher number of markers to be investigated in order to build better diagnostics. Multiplexing immunofluorescence and immunohistochemistry in particular are interesting, as they allow for simultaneous marker detection while keeping the information of morphological context. That is especially relevant in the study of the interaction between the tumor and its microenvironment. So the method chosen to do immunofluorescence multiplexing on LabSat is the pyramid signal amplification, TSA. This technique allows simultaneous marker detection on the same tissue. The working principle is the following. So on your first antigen of interest, you would detect using a primary antibody that you would bind with a secondary antibody conjugated to an enzyme, here horseradish peroxidase, that in turn will catalyze the reaction of tyramide fluorophore conjugate that binds to the tyrosine residues at the antigen site. Before moving to the next marker detection, the antibodies need to be removed by application of heat on the tissue. This step is called elution. The covalent bond between the tyrosine and the tyramide ensures that the signal stays on the tissue, even after the elution step. Then, subsequent markers can be detected using the same method, by applying a different fluorophore. This can be repeated for all the markers in a panel, and once all the tyramide are bound, the slide is ready for imaging. The TSA kit chosen for this project is the widely used Opal kit from Akoya Biosciences. And the goal was to adapt these reagents to the LabSat system. We used a 7-color manual detection for 6 markers plus conscious staining with fluorophores of excitation wavelengths ranging from 520 to 690 nanometers. So our objective is to automatize a full 6-plex using the Opal kit. And while the pre-processing, meaning the deparaffinization, rehydration of the tissue, as well as imaging, will still be done outside LabSat, the machine will take on all the steps that are directly related to the staining itself, starting with the heat-induced epitope retrieval, protein blocking, primary antibody, secondary antibody, and the TSA itself, then elution, all those steps being repeated in a number of times corresponding to the detected markers, and finally contrast staining. The assay development process was organized around 4 main axes. The first one, the single-plex optimization for each marker, with the objective to reach good and repeatable performance for every marker individually. The second axis is the uniformity optimization, with the aim to obtain homogeneous signal levels over the tissue, even over large sections. And then the antibody elution optimization, to make the heat-induced strip-off reach complete antibody removal between cycles while preserving the tissue morphology at the same time. And finally, the multiplexing part, bringing all the individual optimized elements in one multiplexing assay and build a fast and efficient workflow on the machine. The 6 markers selected for the panel are typical immunomarkers: FOXP3, PD-L1, PD-1, CD68, CD8, plus pan-CK for cytokeratins. The optimization of the assay was done on FFPE sections of human tonsils, a positive control tissue for those markers. So first axis of the tonsil optimization project, single-plex optimization. So the assay was directly developed on LabSat with a goal to ensure high staining quality while keeping a reduced execution time. The parameters could be directly adjusted from the software interface of LabSat Research, and the parameters that were available for us to play with were the antigen retrieval, so both temperature and pressure settings, the reagent incubation, here again temperature, duration, and reagent flow optimization, and finally primary antibody and substrate titration, just like in any regular assay. The image acquisition was done on a Mantra imaging station, and the Inform software was used for image analysis. When using the Opal detection system, acquisitions need to be post-processed. In particular, the signal needs to be deconvoluted to separate the information corresponding to each Opal fluorophore. We built a spectral library prior to this study and used Inform to do unmixing, as well as cell segmentation of the tissue for further analysis and quantification. The single-plex staining optimization resulted in bright and specific signal for all the markers with high normalized mean intensity of balanced value across the 6 markers. The results are here displayed in software reconstructed bright field view, which is the informed pathology view. For each marker, the variability of the protocol was assessed on 9 consecutive slides. The single-plex repeatability showed less than 15% of the signal variability, underlining the effective automation of protocols on that system. Now, the uniformity axis. So, uniformity of the fluorescence signal is an important feature of an optimized protocol that can be challenging to obtain sometime, especially on large tissue sections. LabSat offers some interesting functionalities that help obtaining a better uniformity. When using a standard incubation of the reagent on the tissue, depending on the method that is applied, this can lead to an inhomogeneity in the signal level across your tissue. LabSat allows for an active control of the flow during the incubation. The liquid motion in the chamber can be controlled at any time, ensuring a more uniform distribution of the reagent over the tissue. This method, called dynamic incubation, increased significantly the level of uniformity of stainings done on LabSat. We assessed the efficiency of dynamic incubation by looking at the signal gradient on slides incubated in a standard fashion versus the slides that were applying the dynamic incubation method. And we saw that over 1 centimeter of tissue, the signal drop was reduced down to 6% when using dynamic incubation compared to 30% with a standard incubation, which represents a significant improvement in uniformity. The third axis of the project is the elution optimization. The role of the elution step is to ensure a proper removal of the primary secondary antibody complex between 2 marker detections. The method that we use on LabSat is simply applying a high heat to the tissue. To assess the efficiency of this elution method, we compared a reference stained slide versus an eluted slide, which simply consists in applying the elution process after primary antibody and secondary antibody incubations before re-incubating with secondary antibody and the substrate. If the elution is efficient, no signal should be observed on the eluted slide. The elution efficiency was defined as the signal removal rate. This elution efficiency was tested for all the markers in the panel, and signal removal rate was over 99% for all the 6 of them, which was considered as a performant elution. Since our elution process involves high temperature, it was important to test that the repetition of those heat cycles would have limited impact on tissue morphology. So we tested different repetition numbers of those heating cycles for 3 different single-plex, and we could observe no apparent degradation of the resulting staining due to the repetition of the elution. So nuclear morphology, extracellular matrix, and epitope detections were preserved. The last part of this tonsil optimization, it's the full 6-plex assay. And our goal is to build an efficient workflow with a minimum number of user interactions. So the tissue preprocessing and reagent preparation is handled by the user, that then has to load all the elements on the machine. LabSat then perform fully automatically 3 marker detections. Then, due to a limited number of reservoirs, a user intervention is required mid-protocol in order to load the reagents needed for the last 3 cycles. Afterward, the protocol can be resumed and finished. Finally, the slide can be retrieved at the end of the protocol, mounted and imaged. The final result is a semi-automated 6-plex plus counterstaining that can be achieved in 4 hours and 12 minutes. The full assay showed adequate marker detection compared to the single-plex optimization. The conclusion of this first part of the study is that the LabSat offers a TSA-based multiplexing approach with high signal intensity for each marker, an adequate signal uniformity across the tissue section, an efficient antibody elution with over 99% of signal removal for the 6 markers, while preserving the tissue morphology and the epitopes at the same time. All this with a high level of consistency. So we're now moving to the second part of the study, which is the transfer of the multiplexing assay using LabSat on a different tissue type, a cancer tissue. So multiplexing assay optimized on tonsil can sometimes not be completely adapted to other specific tumor sections because of different marker expression patterns, or sometimes tissue specificities like different structures, or different levels of autofluorescence, etc. The question we ask here is this tonsil optimized protocol applicable to my cancer tissue samples? We approach this with a 3-step transfer strategy. First, we applied the tonsil optimized protocol directly to our cancer sample. Then we evaluated the performance and defined what needed to be re-optimized. And finally, we modified protocol parameters in order to reach the desired performance. We're going to go through a small case study on non-small cell lung carcinoma, whole tumor, and TMA sections. Our objective is to reach an acceptable performance on this tissue in a minimum number of optimization steps, because our aim is to avoid the re-optimization from 0. LabSat offers the flexibility of optimization, and some important parameters can be readjusted directly from the software interface, like the antigen retrieval temperature, the incubation times, but also protocols are modular. You can add extra steps like blockings, quenching, etc. And of course, you can always play with the standard element of an assay like reagent titrations. So the first phase of the transfer strategy, we applied the tonsil optimized protocol directly on non-small cell lung carcinoma samples. IHC stainings of the corresponding tissues are shown here as references for marker expression. And we could observe that the direct application of the protocol on this particular tissue showed lower intensity levels than expected for all the markers compared to what we had on tonsil, which indicates that re-optimization might be needed. So we applied consecutively 2 simple protocol adjustments. The first one was to increase the antigen retrieval temperature of the assay, and the second one was to increase OPAL 540 concentration. OPAL 540 is the termite that we associated with CD8. And this was enough to rescue the signal levels and to show appropriate detections for all the markers. We then applied this re-optimized 6-plex to NSCLC tissue microarray in order to assess the protocol on various expression patterns. And here, the re-optimized protocol showed appropriate detection of all the markers on those TMA cores comparably to the standard IHC reference. Various patterns of expression could be detected similarly to the IHC reference using this re-optimized protocol. And the total time of the assay remained unchanged compared to the tonsil optimized protocol, staying at 4 hours and 12 minutes. So as a conclusion, LabSat offers the possibility to run a 6-plex 7-color assay in a timely manner. with high quality results, both in terms of signal, uniformity, strip-off efficiency, and morphology preservation. The tonsil-optimized protocol could be transferred to lung cancer tissue for the same assay duration, and the protocol transfer process was a simple two-step adaptation. Thank you all for attending this webinar. I encourage you to send all the questions you might have to the email address you see below, communication@lunaphore.com. We'll get back to you individually. Thank you and have a good day.